I am going to talk about the origin of life, but you may wonder, how is it possible that we can know anything at all about the origin of life? And perhaps by the time I finish speaking, you'll think, well, we really don't know anything about the origin of life. I'm going to try and persuade you that we can know some things, but it's different to other fields of science. So in physics, for example, if we want to understand the origin of the universe, well, one thing we can do is have a large space telescope like the James Webb Telescope, and we can look to the furthest side of the universe, which is looking back in time, because the further away a star is, the longer it takes the light to arrive, and so we can look back in time and observe directly things that were happening close to 14 billion years ago. That's not something we can do on Earth. We cannot, there is no way of directly observing what the Earth was like 4 billion years ago. There are some rocks, there are fossils, there's bits and pieces, but mostly it's damaged and destroyed, and there's really nothing very much to see. And actually, there's a philosophical point here as well, that if we actually could make a time machine and go back 4 billion years and look for the origin of life, where would we go? And what would we actually look for? And how would we know if we found it? The answer really is, well, we wouldn't. If we found some green slime somewhere that was plainly somewhere between a non-living system and a living system, is that on the direct path to the origin of life, or did it die, or was it never alive? How would we possibly know? Actually, we got the clock wrong on this one. This is only 3.5 billion years ago. And this is a painting I like a lot from Peter Sawyer of the Smithsonian Institution. And this brings the the 5 elements together from this week: the earth, the water, the air, and the fire. And also life, because you're looking at some life there, although not all of you will recognize it. Those kind of lozenge-shaped things in the foreground are stromatolites. There are still a few of them living today in Australia and other places, not very many. And they look a lot like rocks, but in fact, they are living, and you may never have guessed that. If you look at them more closely, they look a lot like rocks. You could almost think of them as a living rock, and if you look at them under the microscope, you see these cells. In fact, they're cyanobacteria, they're photosynthetic bacteria. But they look like sand grains. They're enigmatic. How can we know what they're doing? It's very, very difficult. There's few things in the universe more enigmatic than a cell, because that's the wrong level to look at what life is doing. If we want to understand the origin of life, we need to look under the bonnet and see how that cell is working, what it's doing, what the machinery is, how it's living, how it's processing things in time. And we need to try to understand the principles that underpin all of this and make it simpler and try to see where it connects with the rest of the world, with the 4 other elements, if you like. Well, we look under the bonnet, we look inside cells, and they are shockingly complex. This is why the origin of life seems like such a difficult problem. This is an E. coli bacterium in a beautiful drawing from David Goodsall. Now, a bacterial cell is about 15,000 times smaller than one of your own cells. They're very, very small. And if you look at it even under an electron microscope, you won't see very much inside. And what David Goodsall is doing here is drawing the proteins themselves and the DNA. So if you look at this, the yellow bit at the bottom, that's the DNA, the genetic code in this bacteria. The green thing sticking out of the top, that's a flagella motor. It's a rotating motor that powers, it rotates and it powers the bacteria around. These are extraordinarily mechanical devices that living cells use, even on the smallest possible scale. The little purple blobs that you can see, those are ribosomes. They're the protein-building factories. So if you were to shrink yourself down to the size of, say, a glucose molecule, it's about 150,000 times larger than you are. These are enormous machines, and they make protein. So a protein is basically a long string of amino acids, maybe 200 or 300, all joined up in a long row. Now these ribosomes, they can do about 200 per minute, sticking them all together in a long chain. They're extraordinary machines, and this is the level of machinery that if we want to understand life, we need to understand how all of this stuff arose in the first place. The difference between, say, a bacterial cell that you can't even see with the naked eye and a dinosaur is probably closer in terms of evolutionary distance than the distance between that you're looking at under an electron microscope, if you like, and the origin of life itself. The distance that we must cross from one to the other is a phenomenal distance. There's a huge amount of complexity, even in the simplest of cells. Now that is a ribosome. It's about 2 million Daltons in its mass, which is to say 2 million times the size of a hydrogen atom. This is where all the action is really happening. We're shrinking down now to the size of molecules like glucose. So we're about 15,000 times smaller than the ribosome itself here. What you're seeing, it looks a lot like an electrical wiring diagram or something. It's not actually. This is a metabolic map. All the enzymes, all the proteins in cells, what they're doing is catalyzing a reaction. One molecule changes into another molecule. And each of the little nodes in this is one particular molecule. And the lines connecting them is where an enzyme converts the node above, the molecule above, into the molecule below. So you could think of this as like walking down a street or something, that a car enters a street and there's a blinding flash, and the car, let's say it starts out as a Mini or something, and it changes spontaneously into a Porsche, and then there's a flash and it changes spontaneously into an Audi, and then there's another flash and it changes spontaneously into a tractor. That's bad luck. And it leaves the street as a tractor. That's a metabolic pathway. Stuff comes in at one end and is transformed all the way down this pathway and leaves at the other end. And whatever comes in down at one end of the road always comes in as a Mini, and what leaves in that road always goes out as a tractor. And you can do this individually for each of those pathways. It's an extraordinary thing. There are 1 billion reactions taking place every second in a cell like an E. coli cell. This is the level where life is operating, and this is really, if we're going to understand the origin of life, this is what we need to explain. And you can understand immediately why most biochemists back away and say, okay, we'll study how this system works, but forget about how it started. So, where can we even begin? It seems an overwhelming challenge. But we can always simplify things and think. And one of the most beautiful of thinkers was Christian de Duve. He won the Nobel Prize for discovering lysosomes, one small part of cells. And he, as a lot of Nobel laureates do, go on to think about big questions afterwards, like, for example, the origin of life or consciousness or so on. And for a long time in biology, only people who'd won the Nobel Prize were ever allowed to work on something like the origin of life. It was banned, otherwise you were not good enough. Now things have changed. And they've changed because we can now build a tree of life using gene sequences, and it takes us back very close to the beginning. And so suddenly we can begin, normal people can begin to ask these questions. But I like this quote from Christian de Duve. He says, how did proto-metabolism, so this is a kind of chemistry that's happening in the environment, in some environment somewhere on Earth, how did it come to be replaced by metabolism, this metabolic map that I showed you, or at least a little middle of that metabolic map? He says the obvious answer to this question is that the appearance of catalysts, whether ribozymes, protein, enzymes, or both, was responsible for the transition. So the catalyst is the protein, which is changing one molecule, this molecule, into that molecule. It's catalyzing a reaction, changing one thing into another. We have to ask how catalysts with appropriate properties came to appear. And he says the only scientifically plausible explanation. He, incidentally, was at the other UCL, the Catholic University of Louvain, and was grounded in Catholicism, but was a scientist as well. Very sympathetic, actually, to religion. There's no necessary antagonism, really, between religion and science, and he worked on the origin of life. But, he says, the only scientifically plausible explanation is that the catalysts arose through selection. And enzymes, he says, are selected only if they fit into proto-metabolism. In other words, we've got this kind of chemistry happening in the environment, and what enzymes and the genes that encode them do is speed up this chemistry that was happening in the environment and make it better, make more of it, make it faster, whatever. It's improving what's already there. In other words, there was something there to begin with. And that's a hopeful starting point, because we need to find it. So what I'm going to try and put to you is that life, in fact, is the best guide to its own origin. What we need to do is think about how energy works in cells, how metabolism arose, and why it's structured the way that it is, and where genetic information came from. These are the three big themes, if you like, in how cells work. There's plenty more as well, but these ones I'll try and touch on and talk a little about this evening. So, energy. This is a kind of a scheme of what's happening in you right now. So, if you shrink yourself down and you go into one of your own cells, you could think of this as a mitochondria. It's also, you could think of it as a bacterial cell. The mitochondria is where we're burning food in respiration, so the thing that's keeping you alive. And you can see down at the bottom, it says glucose, burning sugar. We're splitting it down by one of these metabolic pathways to a 3-carbon molecule called pyruvate. And then it goes into this thing called the Krebs cycle. So what's the Krebs cycle doing? In the simplest of terms, it's taking these organic molecules, which are made from carbon, oxygen, and hydrogen only. That's it. And it's pulling out the CO2, and we breathe it out. We're expiring it. And it's pulling out the hydrogen, not as hydrogen gas. We're not kind of breathing hydrogen gas out. But it's taking out the 2 atoms in hydrogen gas, and it's feeding them, and you can see at the top there, it's feeding them into the membrane. And it's passing them on. The hydrogen atom is made of a single electron and a single proton. The electrons are fed to oxygen, and the protons are pumped across the membrane. So what we have, in effect, is a current of electrons to oxygen, coming from food, ultimately. And that current of electrons is powering the pumping of protons across the membrane. So now we have, the protons have got a positive charge, and they're all on this outside of the membrane. And that means there's a positive charge outside relative to the inside, which is negative. So now we have an energized membrane, and this is the membrane inside all of your cells. If you were to kind of iron out all of that membrane in your own cells, you'd have about 4 football pitches worth of membranes. And the charge on those membranes, if you were to kind of shrink down and stand next to that membrane, it's only about 180 millivolts, nearly 200 millivolts, which doesn't seem like very much, but the membrane is very thin. The membrane is something like 5 millionths of a millimeter in thickness, 5 nanometers thick. And what that means is that the field strength, the strength of the electrical field that you experience if you're there, is 30 million volts per meter, which is equivalent to a bolt of lightning. So you've got 4 football pitches worth of membranes with a charge across all of them equivalent to a bolt of lightning. That's what's keeping you alive. That's what we're doing with all our food. So that's how respiration works. And I've got there, this charge, this huge charge on the membrane, is driving ATP synthesis. ATP is often called the universal energy currency, and it's used to make proteins work effectively. A protein, you can think of a protein as a slot machine, and you put your coin in it, and it goes, and you put another coin, and it goes, and that's more or less how it works. You keep on putting money in, spending your ATP, and that's more or less proteins are changing their conformational state to do work, and the work is being driven by splitting the ATP. The ATP is made continuously from the electrical charge on the membrane, and the electrical charge is maintained continually by burning food. So we're burning hydrogen in oxygen, which is in effect burning rocket fuel. So this is the ATP synthase. It's a molecular machine. It's a rotating motor. It's an extraordinary device. And if we're thinking about the origin of life, then, well, this is later. This is something which is a product of genes and natural selection and evolution. This is not something that goes back to the origin of life. But still, this is how the energetics of all cells works. So we need to understand what's a simpler version of this. How can it work in the absence of genes, in the absence of natural selection, and so on? I'll try to explain. This looks exactly the same, or at least it probably does. But there's a few things that have changed here. I'm now talking about metabolism. You'll find this layout in some of the most ancient bacteria that live on Earth. You'll find them in deep-sea hydrothermal vents. You'll find them on land. You'll find them in coal mines. You'll find them in all kinds of places as well. What are they doing here? They've just reversed. The arrows on the Krebs cycle are now going in the opposite direction to the way they were previously. I just said that what the Krebs cycle is doing is taking out CO2, it's taking out hydrogen, we're burning the hydrogen. What's happening here? We're bringing in hydrogen. This could be hydrogen gas. And we're bringing in CO2, and we're making the basic building blocks of all of life.